So hopefully everybody's enjoying their day. It's definitely better than sitting in a small room with teachers, right? That's great. All right. Well, so I want to introduce our keynote speaker. Before I do that, we are broadcasting this via Zoom because we want to use it also as part of our distinguished speaker series that we do within the CTSC. So today, it's my pleasure to introduce very briefly a guy who has done a lot, Dr. Michael Holinstat. He will discuss his very interesting journey in translational research. How did he start? What did he do in his lab? Down to improving the health of people, something that you could see in research. Remember, if you do research in the lab that never translates to humans, it's like a lost time. So his research has been very successful. He's a professor in the departments of pharmacology, medicine, and vascular surgery at the University of Michigan, and is the director for translational research for 2 biotech companies, one in the US and one in Europe. So he's also the director of the NIH CTSI-funded T32 training grant. His research interests focus on understanding lipid, lipoxygenase, and oxygenase regulation of platelet signaling and function. How does it relate to thrombosis, people who have a clot? His work has resulted in numerous patents, not only in the US, China, Japan, Europe, everywhere, for various therapeutic discoveries to treat cardiovascular disease. So it is my pleasure, actually, to introduce Dr. Holinstat. Thank you for coming.
Well, that's quite an introduction. I hope I can live up to it. Thank you very much for having me today. It's really a pleasure, and I've enjoyed all morning seeing some posters, hearing some talks, seeing the 3MT. It's a really great symposium we have here, and I think the addition of or inclusion of high school students is really essential for the future of our field, and so welcome. So I'm going to talk to you today a little bit, very briefly, about my origin. I'm going to talk to you today about how I at least think about approaching a question, how we start, how we acquire what's necessary to know if it's worth moving forward. Once we identify that and do all of our preclinical work, how we then move forward to get it into people. All of which, each one of these steps is part of translation and is essential for its success. So this is the University of Michigan's medical school. It's actually missing a new hospital. We have a new $1 billion hospital. It's about to open in the last green space. So no more sledding in the winter down hills. But it has I just put this picture up to kind of give you a sense of the breadth of research at Michigan. So a little bit about my past. So I come from the Northeast. So I grew up in Connecticut. I did my undergrad at Southern Connecticut State University. And during that time, I really gained an appreciation for science. I moved to Chicago to conduct my graduate work, where I started working in the vessel. It was a basic science lab working on endothelial barrier function. I learned about the endothelium. At that point, and I decided I wanted to think about how I could take advantage of the knowledge I was learning for patient care. I moved to Vanderbilt University to do my postdoc. And at Vanderbilt University, I teamed up, and back in the day, there was no such thing as a CTSA or a CTSC. If you wanted to be translational, you developed it yourself. And so I gained 2 mentors, a chief of cardiology and the chair of pharmacology. And I spent every morning consenting patients in the cardiac PCI lab. Then I would go back to the lab and I would wait. And they had a little monitor on our computer. We could see where the patients were and what therapies or what interventions they were actually going to get. When we found one that was going to get the intervention that I was interested in, I'd run back down to the cath lab. I'd scrub in. Would draw their blood before and after intervention. I'd bring it back up to the lab, and then I would spend the rest of the day processing it in order to answer my clinical question. Okay. Luckily, you have these organizations now, this umbrella CTSA organization that is formalized the training and has bypassed all the hurdles and points where you can fail, so that if you really have a passion for conducting clinical and translational science, there's now a roadmap for its success. And so I'd say take advantage of it. These are my disclosures. I moved, after Vanderbilt, I moved to Thomas Jefferson University for my first faculty position. That was back at the beginning of the Great Recession. So that was the first time we thought the sky was falling and the world was going to end. So those who have concerns about everything that goes on in the world, I would tell you, we go through different things every 5 to 10 years. If you have a passion for what you do, you'll be successful. And don't let anything stop you, right? I spent the early part of my career there. Thomas Jefferson is a very good medical school, and that is highly focused in medical research. I was in hematology, in internal medicine, and in about 2014, word got out that I might be movable, and so Michigan made a hard push to move me to Ann Arbor, which was very interesting to me because I heard about the Midwest. Usually when you fly over it, you kind of look down and say, oh, there's the Midwest. And it's not a slight to the Midwest. It's just people from the Northeast have a very different idea of geography. So we went and visited, and I have to say, I really enjoyed it. I mean, we live in Ann Arbor and love it. My family loves it. I love it. The people are very friendly and are equally as driven and willing to drive success as anyone anywhere in the country. Right. So I've been at Michigan since 2015, so about 10 years now. And we've developed a really vibrant environment. I have a lab of anywhere between 15 to 20 people at a time in the lab, depending on what we're doing. We do everything from ideation through clinical trials. I'm going to give you like a glimpse of that, a couple vignettes of what we've done and what we're doing currently. So the first question I always have to ask myself, if I'm going to study something, I need to know where the gap is. What's the unmet need? If there is no unmet need, why am I studying it? And if there is an unmet need, why is that, and where can I make a difference? Well, cardiovascular disease has no lacking of unmet need. So in 2022, almost 703,000 Americans died of cardiovascular disease. And that number continues to increase. That's one person every five deaths, and that's a death every 33 seconds. So just to give you a perspective on the scope and the need that's currently out there. Disconcertingly, even with all the advances we've made, by 2035, that's only 10 years from now, that's supposed to balloon to 131 million Americans with cardiovascular disease. That's 45% of the population. I want you to think about that for a second. 45% of the population in the next 10 years will be diagnosed with cardiovascular disease. More discerning to me and concerning is the cost to the healthcare. Right? Within the next 10 years, it is going to balloon, or it is projected to balloon. So within the next 10 years, we're expected to move from about $318 billion a year in healthcare costs to treat cardiovascular disease to almost $750 billion a year. Or, to make it sound better, $750 billion a year. So this is a problem that needs to be dealt with. All right, so The rest of my talk is going to be telling you about some of the really innovative ways that we've taken to developing new therapeutics by understanding the unmet need, understanding the targets, and then moving it into the clinic. So to do that, let me just give you a really quick cartoon on the vessel. So what you're looking at here is a vessel. You have red blood cells are in red there. You have the little crinkly things, those are platelets, the most important cell in the body, by the way. And then you have the endothelium lining it. And this provides all the oxygen to your body and all the nutrients. So without blood flow, nothing happens in the body. Under normal conditions, due to insult or injury, inflammation, hundreds of times a day, you're going to have small vascular ruptures or insults, and you're going to end up with blood leakage. You'll hit a wall, you bump into a chair, you get a little bruise. That's what happens. But really quickly, the body is set up to deal with that, and so it forms what we call a platelet plug between platelets and fibrin. And when that happens, the blood is now, that hole is plugged up, blood flows through in the vessel where it's supposed to be, and you continue on your way. You, in fact, never even really know that you had a rupture of a vessel, which sounds really bad. Under pathological conditions, however, such as atherothrombosis, when someone has diabetes and you get an atherosclerotic plaque, when that plaque ruptures, you start with the same hemostatic regulatory plug that forms, but for some reasons we still do not fully understand, the platelets continue to be recruited until they completely occlude that vessel. That's called a thrombotic event. What happens? Once you have a thrombotic event, no blood is flowing, no oxygen is moving, no nutrients are getting to all the tissues of the body, and tissues of the body are dying. Now, in the heart, we call this myocardial infarction, heart death. In the brain, we call this stroke. Okay, so stroke and heart attacks are primarily due to issues with the blood and vessel. So being able to treat those is a first-line therapy for being able to treat all of these other diseases. So we published a paper last year, a state of the field paper, on where does antiplatelet therapy come from. And you might be surprised to know that we had no antiplatelet therapy until the 1980s. Aspirin, you can go to GNC or your local market and buy a bottle of aspirin for a couple dollars, right? You may think that's been around forever. Clinical trials with aspirin didn't occur until the 1980s. Aspirin was not actually a clinical option until the 1980s for treating as a blood thinner to treat platelets and clotting. Quickly after that, we identified epsiximab. So epsiximab was a way to block the integrins on the surface of the platelet. The integrins form the glue that allows the platelets to come together and form that clot. So we can give that IV, and you can block all platelets from activating. That was a major advancement because that was one of the first examples of immune therapy being used in the clinic. Long before it was ever used in cancer therapy, it was used in the blood. We then, you fast forward to 2001, we realized that any one intervention wasn't good enough, so we came up with dual antiplatelet therapy. Well, let's give two. Let's use aspirin, and then let's use a drug that blocks a receptor on the surface of the platelet called the P2Y12 or ADP receptor. So for those of you, for the physicians out there, that's aspirin and Plavix, typically. That's dual antiplatelet therapy. Wasn't approved until 2001. So that's important to realize. These interventions have not been around for a really long time. And then the most recent one was Vorapaxar in 2014, and that was the PAR1 thrombin inhibitor. So as a little vignette, that was a drug that Merck had put out as a triple antiplatelet therapy. That could only be given in patients that were already being given aspirin and Plavix, but were still at risk. That was a 40,000 patient trial, phase 3 trial, the largest clinical trial in history. 81 people died of intracranial hemorrhaging in that trial. The trial failed. The drug was FDA approved with a black box label. The first drug to ever be approved with a black box label. Okay, but still carries a major risk because you're shutting everything down. Okay, so we need to do better. Right? So all those drugs that I just showed you, they only target 4 targets on the platelet. So it really has come to our realization that we don't need more drugs for the existing targets. What we need are new targets. We need more drugs, more targets for the toolbox for the provider to treat patients with special cases. And so combination of therapies may be the way forward, but we can't do it at the risk of bleeding. So we have to develop new therapies that are better at preventing a thrombotic event, that is attenuating but not completely inhibiting platelet activation, but not at the expense of bleeding. Because bleeding is really tough to treat, and bleeding in the brain is extremely difficult to treat. All right, so unlike some other areas of therapeutic intervention, like cancer, where we have historically used poison to kill all the cells, and we hope to kill more tumor cells than non-tumor cells, in the blood field, we think of this more as a pendulum. We can't completely block platelets or you'll bleed. And if we don't block them at all, you'll clot. So it's a very challenging area to make new interventions. And I'm going to tell you a couple of stories about how we've done this. My lab is multimodal. That means we use a lot of different approaches to think about the platelet, to think about its mode of action, to think about thrombosis, and to think about how patients can benefit from this. So I'll tell you today a story about a drug we call ML355. So I know it sounds boring, but pharmaceutical loves to have alphanumeric numbers that tell you where the drug, who owns the drug at the time. So that name will actually change throughout the talk. So that is a 12-LOX inhibitor, 12-lipoxygenase inhibitor, and I'll tell you a little bit about that. I'll also tell you about a newer discovery that takes advantage of a discovery of a new metabolite or oxidized lipid in the blood that one of my graduate students identified that potently regulates platelet activity in a way that's not been druggable previously, and how we've now taken advantage of that to make a drug that targets that and is in late preclinical studies getting ready to move into man. One of the things I won't talk about today, we were the first lab to do the transcriptome of the human platelet. You say, well, okay, who cares? Why would you do that? They don't even have a nucleus. That's because they're special. But it was important because we identified for the first time, we took an observation in the clinic that if you were black, you have a higher chance of having a thrombotic event than if you're white. And the dogma at the time was, well, that's because they don't get as good medical care. Yeah, we can go down the list of stereotypes as to why they said that happened. We said, okay, but why don't we actually do science as opposed to just coming up with answers that have nothing to do with actual science? So we went and recruited 156 subjects, half black, half white, half men, half women, and we identified a single polymorphism. That is one amino acid in one receptor on the human platelet that is highly expressed for people who self-identify as black and lower expression in people who self-identify as white. 83% of people who self-identify as black have this polymorphism, whereas only 30% who are white. And it is a genetic, it is a polymorphism that actually causes 70% of the increased risk. Why is this important? Well, now we can target this receptor because we know that that's the culprit. In fact, Bristol-Myers has a phase 2 clinical trial targeting this receptor because of this. Further, guidelines at the FDA have now changed due to this set of studies. Previously, blood was blood, who cares? Now, if you want to do a study, a clinical study that's going to be approved by the FDA, you must include subjects who self-identify as black and subjects who self-identify as white. You also must include subjects who are female and subjects who are male, because we now know that there are sex differences and we know that there are racial differences, all right, in response. And so in addition to therapeutics, this was one of the first translatable ways we were able to really make an impact in this field. And it is a major impact. So all clinical trials now dealing with the blood have to enroll a diverse population. And then, of course, all great translational science starts with exceptional basic science, or what I like to call ideology. So you have ideation. You think of the new ways. You do experiments. Through experimentation, you identify the best path forward, and then you move forward. Without that step, it is by sheer luck that you will make significant advancements, at least in the therapeutic realm. All right, so my lab has made a career out of this. We started by, identifying, well, there's lots of lipids in the blood, right? And a lot of these are polyunsaturated fatty acids. That is, fatty acids you take in through meat, through plant. You don't make it yourself. You have to intake it, right? So you guys have all heard of, fish oil. That's a polyunsaturated fatty acid. So there's a fatty acid that is highly expressed on the human platelet. It's called arachidonic acid. And traditionally, everybody studied one oxidized lipid from that, and that was thromboxane A2, right? That's why we have aspirin, COX-1 inhibitors. It's to prevent the formation of thromboxane, because there's a receptor on the platelet that thromboxane binds to and further activates the platelet. Okay, great. But look at all these lipids that one fatty acid can make that are in the blood, and we don't even think about them. So I started asking the question, what if one of these other lipids was actually really important? And we looked at 12-LOX, and we looked at it for a very specific reason. We looked at it because only the platelet expresses this enzyme. So immune cells, red blood cells, endothelium, none of them express this enzyme. So if 12-LOX turns out to be an important lipoxygenase that makes these oxylipins that maybe are deleterious to the system, we get immediate selectivity because only the platelet has that enzyme. So blocking it, you're not going to affect endothelial function, immune function, red blood cell function. Okay, so that was our starting point. So I move back and forth seamlessly between human and animal, and I'll try and remember to tell you when I'm talking about human data and when I'm talking about animal data. So we took an animal. Well, we went with the human and said, okay, do humans have 12-LOX? Its gene name's ALOX12. Yes, they do. Do they have the transcript? Yeah, it's in the top 12% of all mRNA transcripts in the human platelet. So it's really highly expressed. Is it important? Unfortunately, when we started looking at this, there are no inhibitors that selectively target it. So we couldn't even ask the question because the platelet doesn't have a nucleus, so we can't knock things in and out easily in the platelet. And we don't have an inhibitor that we can just throw onto the human platelets and say, is this pathway important? So we turned to the animal. We turned to 12-LOX knockout mice. Really quick vignette on that. When we decided to work on 12-LOX knockout mice, just as a proof of principle, the only person in the country that had them was Simon Karpatkin in New York City. And I reached out to Simon, and he had passed away 2 weeks before, and his technician had sacked his entire colony. So, fast forward a year and a half after we re-derived the line. Don't do that. It's a long process. We were able to do this first study. It said just to ask, is it important? So what you're going to look at here, this is what we call in vivo intravital microscopy. So this is an anesthetized animal. We've exposed the vessel. We use a laser. Just think Star Wars. We use a laser to induce a pinpoint inflammation in the vessel. And then we can observe in real time the platelets coming in, forming a clot. They'll be in green. And the fibrin that forms the mesh, and that's in red. And so what you see on the left is a wild-type animal. You see on the right is an animal that doesn't have 12-LOX. So on the left, that's what happens when you have an injury. That's what a clot looks like in your vessel. On the right, that's what it looks like in your vessel when you don't have 12-LOX. You don't form a clot. So that really gave us the first confidence that, okay, this does seem to be a really important enzyme, because if you don't have it, you can't form a stable clot. Fast forward several years, we worked together in a large collaborative with multiple institutions and the NIH to screen over 553,000 compounds in order to identify one compound, that had all the parameters for targeting 12-LOX, but not targeting any other enzyme. and being effective, being able to be used in the blood, being able to cross the membrane to get inside the platelet, not targeting 5 locks, COX-1, COX-2, 15 locks, and so on and so forth. It took a lot of work. But we were able to identify a compound we call ML355. And so we published those first set of papers that really demonstrated that ML355 not only is important for function, but blocking it prevents thrombosis. Really important. Okay. And I always like to use some of these competencies we have over here. These are the NCATS competencies, or as my son likes to say, the merit badges, that really we think about when we think about translational science, right? So it's not just going to your lab and doing something, but are you doing it at speed and efficiency? Are you being creative? Are you thinking outside the box? Are you partnering with other experts? And are you employing team science? Because honestly, big science, that is clinical and translational science, is team science. So you have to be able to do those things, and those are skills in order to be successful. So we brought together a team that involved my lab, a lab from California, a lab from New York, and NCATS in order to get to this point. So I'm going to give you a little bit of crash course on blood biology and how we test it. This is an assay called an aggregometer. It's in every pathology clinic in the world, so if you go to the UH hospitals, you will see one. This is how we determine whether somebody's platelets are acting normally. So platelets are non-adherent cells, so you put them in a cuvette, they're in solution, you have a stir bar at the bottom, and you have Bourne's law, which is you have a light source on one side, a detector on the other. Unfortunately, platelets are non-adherent, so it's cloudy. If you ever take someone's blood and you remove the red blood cells, you see just a cloudy mess, right? When platelets start to activate, they clump, as they're supposed to do, and they fall out of the light source. And that translates in real time to what we call an aggregation or platelet activation. Okay. So we wanted to assess, does ML355 work in human blood? And how does it compare to aspirin? So we take human blood. We gave that we stimulate it with thrombin, which is the most powerful endogenous agonist in the blood. And we either treat it with vehicle or with aspirin. ASA is the acronym for aspirin, or ML355, or both. Do we need both? And what we found was aspirin does what we thought it would do. You give the blood aspirin, you block about 40% of the clot that forms. You give it a 12-LOX inhibitor, ML355, you block 80 to 90% of that clot. And importantly, adding aspirin back in gives no added benefit. So what does that mean translationally? Well, as this moves forward, you don't have to give aspirin. You don't have to think of dual antiplatelet therapy, which increases bleeding risk, because aspirin has no added effect. So that's in washed platelets. So we've removed red blood cells, we've removed the immune cells, but humans don't actually have isolated platelets floating through their blood. They actually have blood, right? So we moved to a whole blood system. This is what we call a flow chamber perfusion system, where we take human blood and we perfuse it through a chamber that has collagen laid down. Why collagen? Collagen is a protein that underlies the endothelium. So when the endothelium, the vessel, is damaged, it is the collagen that the platelets bind to that cause that first activation. So that is the adherence component that we think about when we think about vessel injury. And we do this at arterial shear rates. So these are shear rates of blood flowing through one would see in the coronary artery. And what you'll see, the platelets are labeled in green. So you'll see, again, in real time, platelets adhering. And so we have the whole blood. We split it. We treat half of it as wild type and half of it with ML355. And what you can see is the blood that's treated with ML355, significantly fewer of those platelets are stopping to adhere to that collagen. And how fast is it going? Those platelets have a fraction of a millisecond to decide, am I going to stop? They're not sitting in a petri dish. They're flying through in whole blood at 1,800 per second speed, okay? So that's about 50 to 60 newtons for those of you in high school thinking about, how do I calculate that in my physics? Okay. So it's really fast. So we did that. But then we wanted to expand our team, because it turns out that vessels are not actually straight tubes, right? We like to talk about them that in textbooks, but vessels actually have bifurcations, they have turning points, and all of those are areas where thrombosis and clotting can happen. So we then worked with a colleague of mine who's in Haifa Technion Institute of Technology. He's a director of the Biomechanical Engineering, BME, group there, and he works on developing novel mechanisms for studying the blood. And so we developed what he likes to call the honeycomb, because you have to name something when you develop it. That's one of the perks of being a scientist. And so in the honeycomb, we also see that ML355 prevents the blood, again, this is all human blood, the blood from clotting. So not only a straight tube, but even with bifurcation points, it prevents that adhesion and clotting from occurring. So it seems like in washed platelets, in whole blood perfusion, in bifurcated honeycombs, it seems to work. Okay. So now we turn to the mouse. That was all human work. So now we need what I like to call, so those basic scientists out there, please cover your ears, a complex test tube, not a small person. An animal model is a model. It is not a small person, and very few things you do in an animal will directly translate one-to-one to man, and it's important to understand that when you're doing these studies. But there is information to be gleaned from the experiments, right? Because it's an enclosed, complex system. All right, so in this complex test tube, we call the mouse, we had wild-type mice, and we treated them either with or without ML355. We dosed them twice a day for 2 days, and then we didn't dose them for a day. So almost 24 hours after the final dosing before we induce injury. Now, the type of injury we're doing now, we're not doing Star Wars lasers, but we're actually using a chemical, iron chloride, or ferric chloride, we call it. And ferric chloride denudes the endothelium, or removes the endothelium. So we take a small patch of ferric chloride over the endothelium just for 2 minutes, and that removes the entire endothelium. And that mimics a massive trauma that one might see in a vessel. And what is that going to do? Well, what you'll see on the left in the wild-type mice, you're going to see after the injury, you're going to see them the platelets start to adhere and start to form a clot. And this is a really informative video because this is exactly what an occlusive clot looks like as it forms in the vessel. Right? On the right, you'll see the ML355. You have adhesion, but it's decreased adhesion, and it never moves across the vessel. You never get full occlusion. Now, if you look on the left there and you focus right now, there you go. All flow has stopped. So everything here is dying as the flow is moving down here. All right? That's what an occlusive thrombotic event looks like in real life. Right? So really nasty. Once it occurs, almost impossible to clear unless you clear it mechanically. Okay. So the other aspect that I mentioned earlier, bleeding. Well, if you're too good at what you do, you're going to bleed. Remember I talked about the pendulum. So we do the very sophisticated pharmaceutical grade state-of-the-art experiment. We cut the end of the tail off of a mouse and we see how long it bleeds. That's my very sarcastic way of saying there are no great bleeding assays out there, but this is actually the assay that one does in pharma to detect bleeding risk. And what I would say, and don't worry, it's 5 millimeters at the end of the tail. We're not chopping their tail off. But what this assay can tell us, if the mouse bleeds longer or increased bleeding diathesis, we don't move forward. If it doesn't, I can say mice don't bleed with it. Maybe there's a chance humans don't. And that's, you see how the difference in the strength of the statements I just made? Okay, really important to know. All right, we went back and we said, well, we're not satisfied with that because it turns out humans don't have tails. And so we want to see what internal bleeding looks like. So we looked at the cremastinal arterioles and venules, and we went all Star Wars. But we turned our lasers up, think Death Star now, and we actually punch holes through the vessel. And we're going to measure how long that bleeding occurs. So we add dextrin to the vessel so we can quantify the time of leakage. And what you can see in the venules and arterioles, once the vessel bursts, it takes about the same amount of time for that vessel to seal off. What does that tell us? There probably is not a bleeding diathesis in internal vessels. And again, this is a mouse model. We'll have to wait till we got to humans to see if we actually do have a bleeding diathesis. Now, it's important in every experiment to have a positive-negative control. So we dose some animals with heparin. Heparin is a non-selective anticoagulant, and guess what? Heparin has a high bleeding risk. So, not surprisingly, the mice bleed and bleed. Oh yeah, they continue to bleed. All right, so this paper was an essential study, the first in vivo study to demonstrate that ML355 really could have some potential moving forward. In fact, the American Heart Association, this paper won their Best Thrombosis Study of the Year for potential new therapies. So now what? So you've done all this work, right? And again, I'm fast forward now. It's about 8 to 10 years of worth of work. Okay. So you have to think, if you want to move forward, there are several interests you have to think about. As an academic, your academic interest is identification of important scientific questions. That's the discovery, the ideation. Careful rigor and reproducibility. Does it only work in one person's hands at midnight when no one else is around on a piece of equipment? Well, you might want to rethink that. So we always share our approaches with the community. I want others to do these studies as well in their hands, in their labs. Do they get the same response that we get? And dissemination of discoveries. A discovery made in your lab and not disseminated is no discovery at all. So I encourage my people, go out and talk about it. Go to meetings, talk, give posters, give 3-minute thesis, publications. The only way the scientific community advances towards clinical development and new therapies is by being on the back of each other and making those continual, seminal, important discoveries that move that needle closer. All right, so that's the academic side. But there is the industry interest. So what's the industry thinking about? So you flip it on. If I'm a company, what do I think about? Well, commercial potential, target product profile, development, investment profile, market potential, and my exit strategy. So if you're going to translate your findings to get to the clinic, and you do need to partner with industry if you want to get to the clinic, I don't want you to think you can do this with NIH money. You can't. It costs too much. So the Vorapaxar study I talked about at the very beginning, that phase 3 study with 40,000 patients, that one clinical trial cost over $1 billion. Right? So if you really want to move a therapy into the clinic, you partner. But you have to understand what industry's interests are, and your interests have to be able to align with theirs. Right? So, fast forward, everything looked, and preclinical looked good. We formed a company. In fact, the COO and CSO of the company came from NCATS, from their drug discovery lead arms. They actually have a whole screening facility in Rockville, Maryland, and will screen your compounds and help you work out compound screening for free as a collaborative. We brought in a CEO. I'm the Vice President of Translational Research, and we brought in a scientific advisory board. Oops. Yeah. I put this slide up to tell you, to really drive home, it is a team sport, or as we like to say, it takes a village, right? So you need financial backing. So you can see what goes into financial backing for bringing a drug forward. So we have Sanofi. Most of you have heard of Sanofi. They're a big pharma. We also have a number of venture capitalists, VC groups, from all over the Americas, from the U.S., from Canada. We also have local funding from the Alexandria Ventures, the Maryland Momentum Fund, and we have the JDRF. So foundation gets involved. So all of this is important if you want to drive something forward. It's also important that you own something. There's got to be something from the company's perspective that they can sell at the end of the day. So if you don't patent what you find, it's important information, but it's information that anybody can take advantage of. So why would a company invest $800 million, $1 billion if they can't own it at the end of the day? So you have to patent. That's part of the process. And for us, since there were multiple institutes that were involved in the discovery, more complex. It takes a village. We have to work together on the patenting and licensing that is put forth to industry. So there's a lot of complexity here, right? And we could have whole lectures on each one of these, but I just want to give you a sense, a glimpse of where we are with that. Okay. So then you need a disease. So I could have gone after arterial thrombosis. I mean, some of our papers demonstrated it was beneficial. It turns out, for whatever reason, professors don't have billion-dollar bank accounts. So if you know someone that does, please have them contact me. So we went after a rare disease, heparin-induced thrombocytopenia and thrombosis. Why did we go after that? Well, patients that receive heparin often will be diagnosed with thrombocytopenia through the 4T score. They will then be given the only FDA-approved drug, argatroban, which is a direct thrombin inhibitor. It doesn't actually even target the platelet. It's a platelet disease. I want to emphasize, it's a platelet disease. But the only drug that's actually FDA-approved is not a platelet drug. They'll put them on argatroban in the hospital. They'll wean them off of that, give them warfarin for 30 to 90 days, and wean them off of that. Okay, great. To give you a further background, about 12 million Americans every year get heparin for surgery, for DVT, for other procedures, and about 1 to 3% of those patients are going to get hit, or at least suspected hit. And of that, at least 300,000 of those people will have confirmed hit, right? And again, the only standard of care is argatroban. So what happens when you treat someone with argatroban for hit? Well, about 20% will have a major bleed. About 25 to 30% of those patients will die. About another 10% will have gangrene amputation. And another 20% will have a new thrombotic event. I don't know about you. That doesn't sound like a great approach, right? So here's your unmet need. We can make major inroads in this because we had tested the only immune receptor on the platelet. This is an immune disease. The way the disease works is that platelet factor 4, a protein in the platelet, gets released into the blood. Heparin, which is the most negative thing you can ever put into your body, and platelet factor 4, which is positive, well, they come together. Charge, negative and positive. And for some people, they have an antibody that exists in their blood. It's a pre-existing antibody. We don't know why some people have this. And it will interact with the heparin platelet factor 4 to make what we call the HIT complex. That will then bind to the only immune receptor on the platelet and cause the platelets to be activated, consumed, so you clot and bleed at the same time. Do I treat the bleeding or do I treat the clotting? You're doing both. That's why it's such a bad disease. All right. So we developed ML355 to address that. So we did turn to a mouse model. Problem with a mouse model, remember I said mice aren't small people? Mouse platelets don't actually have an immune receptor. There's a problem. So we had to humanize the mouse. We actually added the human immune receptor to the mouse platelet. And so we have humanized mice that are humanized for the potential for HIT. We cross them with mice that don't have 12 locks, right? And so what we are looking at is thrombocytopenia and thrombosis. And what you can see in the top graph, we're looking at platelet counts. So that's thrombocytopenia. So that's thrombocytopenia is low platelets. That's what it means. It's a really fancy clinical term. It just means low platelets. All right. So if the animal didn't get the Fc gamma, so if they weren't humanized, nothing happens when you induce that system, when we inject an antibody that causes HIT to occur. If the animals do have that receptor, within 30 minutes, they lose about 80% of their platelets in their body. If we treat them with 12 LOX inhibitor, or if we cross them with an animal that doesn't have 12 LOX, we can prevent thrombocytopenia from occurring. Now, the other part of HIT is thrombosis. So what you're looking at here is the platelets are labeled, and 4 hours after the induction, we sack the animals and we look at their lungs, say, do they have clots? Well, the vehicle control lights up like a Christmas tree. Lots of clots that were formed. The animal that was treated with ML355, you've completely prevented thrombosis. And the animal that doesn't have 12 locks, you've completely prevented thrombosis. This was the animal study that the FDA needed to see. That's why I'm showing you this model. They needed to see this to give us a thumbs up to move into man. All right. The final study we did was we did a cryo-EM. Nobody had ever been able to crystallize or cryo-EM the enzyme. So we did that's why it took us 553,000 compounds to find one that worked. So we were able to, with that compound, to actually, for the first time, do a cryo-EM of the enzyme and demonstrate where the drug fits into the enzyme in the pocket that we had predicted it would, but for the first time we could demonstrate that. All right. So where are we with this first vignette? Well, arachidonic acid, which is highly prevalent in the platelet, can be blocked by ML355. It's now called VLX-1005. Remember I said alphanumeric? So Verilox Therapeutics is the company I started, so we renamed it. That can prevent platelet activation. It took us, from initiation in 2010, took us about 4 years to develop ML355. Took another 4 years to do preclinical. Then we licensed it in 2018, 2020. We received orphan drug designation by the FDA. That's the designation the FDA gives because there's no treatment right now. And so to reward a company that's willing to go after an orphan disease, they will give them protection when it gets marketed. And that's ODD, if you hear that word. Then we got IND approval in 2020, finished our phase 1. 2022, we received fast track designation from the FDA. That's a special designation that where we've demonstrated this is a dire need in the clinic. That patients are dying because they're not adequately treated. And so with fast track designation, the FDA has a specialized team that is dedicated to working with you to move this through the clinic. This is currently now being given to patients with HIT. And hopefully by next year, we will have finished our phase 2 to move into registrational phase 3. And as a rare disease, luckily we don't have to do 40,000 patients. So that's where we are with my first vignette. What we learned from that, however, was how important the enzyme is. So it turns out that 12-LOX, or 12-lipoxygenase, actually oxidizes lots of fatty acids, not just arachidonic acid, to make lots of oxidized lipids. And so we've published a number of papers demonstrating how each of these is important to regulating blood and blood function. But in particular, I'm going to focus on one, dihomogammalinolenic acid. Yeah, I know. You can unglaze your eyes. It's over. The pain's done. We'll call it DGLA. It's a fatty acid that is the third most expressed fatty acid on the human platelet. It's also highly enriched when you take supplementation and certain foods. And so what we showed was back in 2016 was, well, if you have this DGLA and it interacts with 12-LOX in the platelet, you get a oxylipin called 12-hetra, and that 12-hetra is an antiplatelet agent. It's an endogenous antiplatelet agent. This is one that the body makes. And so we can show here that if we give 12-hetra to human blood and you stimulate the blood with collagen, you attenuate the ability of those platelets to become active. If we take an animal and we give it DGLA, a wild-type animal, you can see you attenuate the aggregation. But if the animal doesn't have 12-LOX, you lose all that protection, which suggested and supported that it really is the 12-hetra that's doing this. So then we said, well, okay, that's great, but since we have the tools and technology, let's look in vivo. So we took animals and we dosed them either with DGLA or 12-hetra, and we looked at their vessels. We induced an injury, and what you can see is both DGLA and 12-hetra attenuate clot formation. Again, these are the early stages of, is this something, we make a discovery. Is that discovery important? How does that discovery work? You have to go through the methodical stages when you want to develop something that's going to go into the clinic and help patients. All right, so a lot of hard work from a graduate student identified the signaling pathway for which this mechanism is working. And it turns out it's working through a GS-coupled GPCR, G-protein coupled receptor, and it works because it causes the cell to form cyclic AMP. And cyclic AMP in the cell, in most cells, activates. That's why it's called GPCR. But remember, the platelet is not like every other cell. It's special. So in the platelet, when you form cyclic AMP, you actually inhibit. All right, this is why phosphodiesterase inhibitors work in platelet intervention. All right, so we had to ask, well, is 12-hetra selective for the IP receptor, which, by the way, was a hypothesis at the time, or is it one of the other GS-coupled GPCRs in the platelet? So we use pharmacologically, this is all human blood, pharmacologically, if we block the EP2 and EP4 receptors, that has no effect on the inhibition of platelet aggregation with 12-hetra. If we block the DP1 receptor, which prostaglandin D signals through, that doesn't affect it. But if we block the IP receptor, we completely reverse all the inhibition. So it does seem to be the IP receptor. So that's the model that we came forward with. We then worked with Garrett Fitzgerald. Some of you may know he's at University of Pennsylvania. He's the world's expert on prostaglandins. In fact, that first slide I showed with the timeline of drug therapy, he did the first clinical trials in aspirin to demonstrate aspirin was an effective blood thinner. So he gave us the IP receptor, and we can see in an animal, now these are animal studies, where you don't have an IP receptor, you don't have any inhibition of platelet aggregation. So now what? Well, academic interest. Let's come back to our what do I think and what do they think. So new ways to regulate clotting. Oh, that's pretty cool, right? We identified a new tool. Somebody at the break had asked me, so clinical and translational research isn't really mechanism, it's just implementation. And my answer was, no, you need to identify new mechanism of action to really advanced implementation in the clinic. High selectivity. Potential for better clinical outcomes. What is industry thinking about this? New patents, orphan drug designation, expand the company pipeline, increase the preclinical portfolio, increase the market potential, increase the company value. All right, so if you want to work with industry and translate your work, you have to understand what is important to them, as well as what is important to you, and it has to align. So I worked with a very talented industry medicinal chemist. So another vignette. You may not realize Lipitor was developed in Ann Arbor. So Pfizer was in Ann Arbor, had a huge facility. They developed Lipitor, and then when they pulled out of Ann Arbor, the University of Michigan acquired their medicinal chemistry group. So we have, at our fingertips, a whole team of industry medicinal chemists. So I took advantage of that and worked with one of the discoverers of Lipitor, and I said, well, I've got this thing called 12-hetra. The body makes it, which is great. We know it's signaling, which is great, but I can't really use this to make a drug because it already exists. I need you to make me something like this, but not this. And he took out a scratch pad and actually scratched a couple of structures together. So I bet it's one of these. So we didn't have to use 553,000 compounds. We used a very talented medicinal chemist. And with that, we were able to, and it wasn't one compound he made, it was a series, we were able to develop CS585, which was originally called OxyProtect, a much cooler name. Oxylipin Protect, OxyProtect. It was commercialized, so we patented it, and we licensed it to a company called Cereno Scientific, bring in Europe. This is a Swedish company. So Cereno Scientific, CS585. Okay, so the compound is now CS585. It's been licensed. Does it work? Well, we looked at all the endogenous agonists that activate platelets and said, well, if we give CS585, what's the dose dependency? How potent is this drug? Well, thrombin, collagen, and ADP all have a potency in the very low nanomolar range. This is really great. This means you don't have to give a lot of drug to get an effect. So then we went and we looked at the human platelets and we said, well, okay, but beyond platelet aggregation, does it affect the integrins? Remember I said those are the proteins that cause the glue for the platelets. And so And does it affect granule secretion? So there's two types of granules in the platelet, dense granules and alpha granules. They both play different roles. Happy to go into that more later on if someone has a question about that. So with the integrins, luckily, we have an antibody in humans that only binds to active alpha-2b beta-3, which is the integrin on the platelet. And we have antibodies that bind to p-selectin, which is released from the alpha granules, and CD63, which is released from dense granules. Why am I telling you all this? Because obviously I'm about to tell you we did some dose response curves to see whether these aspects of the platelet were affected. And you can see at the low nanomolar range, we significantly attenuate integrin, alpha granule secretion, and dense granule secretion. We then wanted to identify if it really is going through GS, we should be increasing cyclic AMP. So we measured cyclic AMP directly. And at low nanomolar range, we can get a significant amount of cyclic AMP formed in the platelet, almost as much as what is formed when you directly activate an enzyme, adenyl cyclase, that makes the cyclic AMP. So very potent, highly activatable. There is a possibility, however, that was brought to our attention, well, maybe you're really regulating cyclic GMP. So we tested that. And our drug does nothing to cyclic GMP. So negative results are as important as positive results because we're looking for selectivity, especially because we want to give this to people. And each one of these has different effects in different tissues in the body. So you're not thinking about the blood you took out of the person. You have to think about every organ in their body. And this drug, once it's on board, has the potential to affect all of those. All right, so then we looked at, so cyclic AMP causes activation of an enzyme called protein kinase A, or PKA, which then causes phosphorylation of proteins, right? And one of those is VASP. And so you can see here at picomolar concentrations in human blood, CS585 can cause VASP phosphorylation. It also is completely blocked when we block the enzyme PKA. So we know it's going through this pathway. Again, all these experiments are meant to demonstrate pathway selectivity because if you want to go into man, you need to know what it's affecting because you need to know what its, not only its intended uses are, but what are its off-target uses or effects. So we wanted to go and look at selectivity. So we look at VASP phosphorylation. If we block the DP1 receptor, it has no effect. If we block the EP2 and EP4, it has no effect. If we block the IP receptor, you lose all VASP phosphorylation. So that makes sense. We then went to a genetic model, so now we've moved back to animal to say, okay, pharmacologically it looks selective, but is it really? So let's use IP knockout animals. What you can see if we give forskolin, it doesn't matter if you have the IP receptor, you get fast phosphorylation. However, if you give CS585, you only get fast phosphorylation if you have the IP receptor. Again, genetically confirming the selectivity. All right, now we come back to the cremaster, and what we see in wild-type animals, if we give CS585, you completely block clot formation. In animals that don't have the IP receptor, clot formation looks exactly the same with or without the drug. So in a clot perspective, in vivo perspective, it is the IP receptor that does everything. Why does this matter? Well, it turns out there are other IP receptors that are in clinical use, iloprost and selexipag. Iloprost is an awesome drug if you want to take something 9 times a day, only by aerosol, and by the way, you won't be protected at night, because that's what the label says, because it has a 30-minute half-life. Selexipag is slightly better. You can take it orally, and works for 4 to 5 hours. You only have to take it twice a day. But it's also much less potent and has a lot of off-target effects. So the question we asked is, these were all designed as IP receptor agonists, like our drug. Do they function like that? Well, I just demonstrated in the IP knockout mouse you lose all effect with CS585. Turns out with iloprost you don't. Iloprost actually not only activates the IP receptor, but a whole bunch of other receptors. This is what causes, for those of you who treat pulmonary arterial hypertension and use iloprost, this is what we call the off-target effects that limit patient tolerability. It causes GI effects, it causes neurological effects, and so it's a drug that's hard to keep patients on. And say, okay, well, what about selexipag? That's the newer drug, meant to replace iloprost. Well, guess what? Selexipag also is not selective to the IP receptor. So we then finally look at human blood and look at aggregometry, and we can see that you block these other receptors, you have no effect. If you block the IP receptor pharmacologically in human blood, you completely block platelet activation. And so we then do the flow chamber, and what you can see in flow chamber, remember this is arterial shear, this is human blood at the shear rate of what goes through the coronary artery. And you can see that at CS585, you significantly decrease the amount of platelets that will adhere, but they're not eliminated, so we don't have the bleeding risk. We can show that it's dose dependent, and we can show that in the bottom row, if we add an IP receptor inhibitor, we completely block the effect we see with CS585. We then dose the animals, dose dependency. Well, how low can we go? And what we can see is we can go down to less than a mg per kg dosing, which means for a patient, you divide that mg per kg by 12.3. That's the generally accepted metabolism difference for drug utility. So you're going to be at well below a mg per kg in human. We looked at at dosing by IV. And by IV, this drug works for over 18 hours, a single administration. We looked at oral. A single oral administration was effective at blocking clotting for over 24 hours, but reversed within 48 hours, so it's not irreversible. It doesn't cause bleeding in the mouse tail, so that's good. And so conclusions. Successfully translating discoveries to the clinic requires several things that go beyond just experimentation and technique. Team building, boundary crossing, addressing unmet needs, collaboration, partnership with the university, industry, government, all of the above, and new patents. You have to have something to actually put in the clinic, right? So these are what I call the soft skills, equally, if not even more important than the techniques you're going to do. And so we now know that we can use ML355 VLX 105, but we can also use CS585 to regulate clotting. So we now have two new tools, not one, but two new tools to go in our toolbox. And I just show a comparison of clinical development. And you can see CS585, I'd like to say, we learned. The more you do, the more you learn. So at CS585, we discovered it in 2014. We initiated drug development in 2018. I literally shelved the drug for 2 years until I decided to work with industry. And in 2020, we licensed it to Sereno Scientific, and it's in late preclinical development. We're actually looking to go into first in man by 2026. So we are fast-tracked, and we're on our way to get this into patients in the 2020s. And with that, I gotta be honest, I couldn't do any of this. I just sit up here and give funny talks. It's the lab that does all the work. These are the people. I just want to demonstrate we have everybody working in our lab, from undergraduates to graduate students, postdocs, research faculty, phlebotomists, clinical coordinators, technicians. It takes a team. And that's not even including the physicians and the patients and the subjects that were kind enough to give us blood, and all of our collaborators from around the world. Sweden, Israel, Australia, even as far as California. And then of course our funding. It's not just NIH, it's not just industry, it's foundation. We have a state funding program for commercialization that is really helpful for things like, let's make a drug. They'll give you the money to turn your idea into a drug, right? So those early stage funding initiatives pay off dividends. And so with that, I'm happy to take questions. Thank you very much.
If there are any questions, you can head to the microphones.
So clear.
Here we go.
Hi. So at the start of your talk, you mentioned looking at like
it's unknown why platelets continue to, during a thrombic event, platelets continue to like pile on. Do you think that's an area where like research could happen? Because that kind of stuck out to me as something interesting.
Oh, absolutely. We still don't know the shift from physiological to pathophysiological that shifts us from a hemostatic plug that we need to a thrombotic event, which nobody wants. So it is a very active field, and I think it's a field that needs more ideation research.
Thank you.
Yeah.
Hi, Mike. If you could start it all over again, what would you do differently?
Well, it's a different world we live in, as I kind of brought up. I love what I do. If it doesn't come across when I give my talks, I'd be surprised. I have a passion for being able to benefit patients in a way that is not one patient at a time. I want to benefit thousands, if not hundreds of thousands of patients, and I love what I do. What I would say is building teams early, networking early, going to more conferences as an early stage trainee, and actually asking questions, getting up and introducing yourself to somebody. That is an area I don't think we do as good a job. We try, but I just think that getting people over that early shock of, I can't talk to them, they're an expert. The way that the people who end up, a lot of people who end up in my lab because they come up and approach me at a meeting or a conference or an event like this and say, wow, that's really cool. I would love to be able to do that. You'd be surprised how many of those experts will invite you into their lab and will sit down and have that conversation with you. And if it's industry, call somebody up. They'll pick up the phone. And that's an area I think had I known then what I know now, I would have done a lot more of that at earlier stages.
Right.
Yeah.
I think that is kind of the key to the whole day, right? It's about making connections and networking, and you never know who knows somebody.
Yeah,
So let's give a round of applause for Dr. Holinstat.